Good afternoon, everyone. As you can probably tell, I'm not Cheryl Walker. My name is Bill Farland. I'm the chair of the standing committee and have had the privilege to do this for the last number of years. We've had an opportunity to have a very good start this morning, I think, for this particular microbiome workshop, a continuing theme among the workshops that we've done. The session this afternoon that we'll begin with, what does the microbiome contribute to inter-individual variability and susceptibility to environmental exposures? Inter-individual variability is another theme that has been recurrent among the workshops that we've done. We've actually done two of those, the last one just at the beginning of October this year. And in those particular cases, we were talking about this from the standpoint of the various activities or characteristics that make up inter-individual variability. And we talked about heritable characteristics and stress and body weight, genetics, all of these that influence responses to environmental stressors. And certainly the microbiome came up as a potential modulator. But this afternoon, we're going to have several speakers who are going to address that issue directly, and then we'll have a session following this that will allow us to talk about some of these and take some of your questions around this issue of inter-individual variability and susceptibility and the role of the microbiome. So without any further ado, I think we'll get started. And the first speaker this afternoon is actually coming through our webcast. And so we'll be seeing him on the screen shortly. The title of his talk is Human Microbial Communities and Variability in Response to Nutrition. Eran Elinav is from the Weizmann Institute of Science, and we're very pleased to have him join us through the modern technologies that are available to us that we hope will not fail us. So, Eran, can you hear us?
Yes, I can. I hear you very well. Do you see my presentation?
We do. We see the first slide up there, and we're ready to go.
All right. So thank you very much for inviting me. This is my very first ever transatlantic presentation, so I hope things will run smoothly. My laboratory here at the Weizmann Institute of Science in Israel studies the microbiome and its interactions with the host, and we regard the microbiome as a signaling hub that integrates signals coming from the host, including its genetics and immune system, but also a variety of factors that affect us as part of the environment. All of these factors integrate and are processed by the microbiome, which lead to very diverse and poorly understood interactions with the host that determine our homeostasis. And when we believe that when these interactions go wrong, then propensity for a number of multifactorial diseases develops, and we study many of these at our lab. But today I will be focusing on one particular group of diseases which are very familiar to all of you, which is obesity and its complications. And as you know very well, obesity constitutes one of the most severe epidemics in human history. The numbers are just staggering, and together with obesity, we are experiencing a great emergence of many closely associated diseases, in particular type 2 diabetes, which today is estimated to affect close to 500 million people worldwide. And interestingly enough, many of the factors which we study and believe are affecting our microbiome are the same exact risk factors that affect our propensity and risk to develop obesity and its metabolic complications. And today we'll be focusing on one of these factors which we study, which is our diet or our nutritional status. And all of the things I'm going to tell you are done in close collaboration with a good friend and colleague of mine, also from here at the Weizmann, Eran, who is a mathematician working closely with us. We've recently contributed to the field by studying how what we eat affects our microbiome and our metabolic homeostasis. We particularly focused on a group of dietary compounds which are consumed by hundreds of millions of people worldwide, and these are non-caloric artificial sweeteners. We've studied a couple of them, and in a variety of mouse models and human studies, we've shown that in some cases, in some microbiome configurations, some of these artificial sweeteners are indeed inert to the host, and this is why they're non-caloric, but they're not inert to our microbiome and can modulate some microbiome configurations in a way that could lead actually to a propensity to develop glucose intolerance, which is counterintuitive because many of us are taking these compounds to counteract these exact set of disorders. But these studies have now been corroborated in mice, in rats, in piglets, and recently several of our major observations were seen in 3 prospective long-term human studies from different places, including North America. But what was really interesting to us is that it is not only what we eat that affects our microbiome and our metabolism, but also when we eat. And in another study which we published a few months ago, we discovered that our gut microbiome features a very robust circadian or diurnal rhythmicity manifesting in many different levels, in the compositional level, in the functional and gene pathway and module content level, and also in the metabolite secretion level. We've discovered actually that this microbial circadian rhythmicity is tightly regulated by the host, both by the host circadian clock machinery, which is present in every single cell in our body, but also, and this is the major regulator, this microbial circadian rhythmic behavior is regulated by our timing of feeding or by our feeding rhythmicity. And this is physiologically important because what we've seen both in animal models or in humans that suffer of severe circadian disturbance such as jet lag or shift work, that one of the first and striking things that happen is that our microbial circadian rhythmicity is disturbed, and this leads to a very profound risk, which has been noted as far as 50 years ago, to develop obesity, diabetes, and their complications. And based on these studies and many studies coming from our colleagues, we are now starting to look at the homeostasis in the gut in a different way than we've looked at it before, not as a static homeostasis, but actually as a kinetic evolving system in which the human side and the microbiome constantly change in a diurnal fashion, in a fascinating and poorly understood tango. And this coevolution of diurnal variation actually determines our homeostasis and is regulated both by our food intake, what we eat, when we eat, as well as by our central and peripheral circadian clock machinery, and we and others are extensively studying this concept. But what I want to talk to you in the next few minutes are insights that are gained from all of these discoveries that were recently made in terms of human nutrition and how it can affect our metabolism. And we've asked ourselves a very naive question: can we use all of these new insights on how nutrition modulates the microbiome and modify nutrition as means of altering both the microbiome and the downstream metabolic risks? And if you think about it, many of us around the world are trying to modify our nutrition and our metabolism by testing and trying a whole lot of different diets. And the sad truth is that most of us miserably fail, and this is not just what we think. This has been studied extensively in large-scale studies, such as this study recently published by the British government. And after a year or two, most dieters miserably fail and return to their original weight or even to higher weight than they started. And we've asked ourselves, why are we so miserably failing in these global dieting attempts? So this made me read very boring old nutritional studies, and it really came to my mind that I understood something that I did not realize before, which is that our entire dietary paradigm is based on a set of scoring system or grading system which give grades to each of our foods or each food component that we eat. And these scoring systems can be calories and they can be glycemic index, but if you go to a doctor or to a dietitian and prescribe yourself a diet, you are very likely to be using one of these food grading systems. So if we take, for example, the most commonly used food grading system around, which is the glycemic index, this is based on old small-scale human studies from the end of the 70s and the beginning of the 80s, in which very small groups of humans, usually 10 to 15 humans, were given an identical food, and their blood sugar levels were followed for 2 hours after they consumed this food. So, for example, if 10 of these people consumed an identical called celery, their blood sugar levels were measured after 2 hours, and the average of these 10 people was given a number, and this is the score given as the glycemic index of celery. But if the same group of people would be eating a chocolate cake, then their average rise in sugar levels would be higher, and the average rising glucose levels in these individuals would be the number that would be given as the glycemic index of chocolate cake. And if you look in your smartphone over the internet, you will find these huge charts that give a score or a glycemic index to almost any food you can conceive, and most of the diets that you ever tried are based in one way or another on these scoring systems. Now here comes the big problem. When you do this drill, not on 10 people, but on 1,000 people, like we've done in the study I'm about to share with you, you can see that the average glycemic response of the 1,000 people is indeed the glycemic index. But if you look at the individual responses, they greatly, greatly vary. And you can see here that when we gave the 1,000 people an identical amount of glucose, you can see that some people spiked greatly after 2 hours on this glucose, while others didn't react at all. And when we did the same with an identical piece of white bread, you can see exactly the same phenomenon. And actually, we've tried this with tens of different foods, and we always got a very great variability in people's individualized response, which by definition already tells us that this concept that we all believed in for so many years of a one-size-fits-all diet cannot be effective by definition. And what we believe is that our dietary paradigm must be shifted to one that is tailored to the individual. Now, why am I telling you all of this in a microbiome workshop? I'm telling it to you because in 2015, we're in a very lucky period in which for the first time we can very accurately quantify the major factors which affect our variability or our variable response to metabolic factors such as our glycemic responses. And these include our host genetics, which are very important, our lifestyle, but the most interesting and poorly studied factor affecting our variability of them all is our gut microbes. And this was the basis for a project which we started 3.5 years ago, headed by 3 talented graduate students, which we call the Personalized Nutrition Project. And in this project, we thought that if we could enroll a large cohort of healthy human individuals and basically collect as much personalized information from them as possible for a limited amount of time, we could try and use very advanced computational models based on machine learning to try and devise personally tailored diets that would predict their individualized glycemic responses to unlimited numbers of food. And this is how the study looked like. This was an internet-based study. People registered online. And then when their turn came, we basically asked them to give us a week of their lives, and we collected from them really an unprecedented amount of personalized data. They filled many questionnaires related to background medical history, to dietary preferences, and many more details. We took blood tests from these individuals. We profiled their host genetics by a million SNP analysis. We connected each individual for an entire week to a continuous glucose monitor, which measures their blood sugar levels every 5 minutes for an entire week, which gives us over 2,000 measurements of blood sugar levels from each individual. And of course, we sequence and analyze their gut microbiome very deeply, both by 16S analysis and shotgun metagenomic sequencing. Throughout the week, people used a smartphone app that we developed for this study, in which they recorded everything that they did during the week, what they ate, how much they ate, when they went to sleep, when they woke up, and so on and so forth. And already after a week, people got a very interesting mirror image of themselves, and this is always very surprising. People are really not realizing who they are and how they behave in real life. We also, gave all the participants an comprehensive analysis of their gut microbes, which is also always very interesting to people. But the most important thing that happened is what happened after the week. A very large and smart group of computational biologists at the Weizmann Institute took all of these huge amounts of personalized metadata and devised machine learning algorithms for each of the participants that are meant to predict that individual's response to different foods, including foods that they haven't consumed during the week. So during this large-scale study, we profiled 1,000 people. We measured over 50,000 meals. We've performed over 2 million glucose measurements. We sequenced over 10 billion metagenome reads. And this study, although it was not advertised, became viral, and we have 5,000 people waiting in line. When we looked at the population cohort statistics, we were happy to see that our statistics were very similar to any other developed country, with over 50% of people being overweight, 20% being frankly obese, and with 25% of people being pre-diabetic, and we'll return to these in a little while. We also were pleasantly surprised by the level of compliance, which is also always an issue with these studies. And you can see here one of the measurements we made by comparing what people filled in their dietary questionnaires before the study and how this compared to their online dietary logging during the week. And you can see that the agreement was extremely good. And what we believe is that people intuitively understood that the better data they give us about themselves, the more we can give them back. And we profiled very deeply the microbiome, and and what we saw is that when comparing to the other big microbiome databases, the MetaHit from Europe and the HMP from the US, you can see that generally there are some differences, but in general, the microbiome in our population was very similar to the European and the American microbiome configurations. Another sanity check, which was very good to have, is comparing the glycemic response in our cohort to what we expect from previous study to be associated with it. So you can see that the higher BMI people have, the higher their glycemic response is. You can see that the higher people have an hemoglobin A1C, so they're more prone to diabetes people have, the higher their glycemic response. The same is true for wake-up glucose levels. The older people are, the higher their glycemic response. And we found an inverse correlation between the good HDL cholesterol and the glycemic response. So all these were sanity checks for our cohort. The only intervention which we made during the week was a breakfast after a night fast that we provided to individuals, to all the 1,000 individuals. This was an identical breakfast consisting in 2 days with identical pieces of bread, 2 days with bread and butter, and 2 days with glucose, and 1 day with fructose. And this allows us to collect really a lot of data and to actually compare this cohort, at least in this one time point. And you can see that when we gave the test foods to these individuals, as I've shown you before, the average response, the average glycemic response of the cohort to each of these test meals was indeed the glycemic index, but you can see for each of them how big, how huge the variability is in the individualized response. And since we gave each of these test food twice to each individual, you can see that while the inter-individual variability is huge, the intra-individual variability, in other words, when a person gets the same food twice, is very small, and so the numbers that we get per person are very consistent when repeated. And this huge amount of collection gave us a lot of data, and you can see here an example of the data collected just for the test foods, and we'll be analyzing these for a long period of time, but it starts to give us a lot of interesting insights. Just as an example, you can see, again, as a sanity check, that the more diabetic and obese people are, the higher the response to all the test foods, so whatever food you give them, they have an average higher response. But then you can start seeing interesting and unexpected associations for some foods and not for others. For example, the phosphotransferase system in bacteria, which is the carbohydrate internalization system, is correlated with fructose but not with other breads, and you can see some families of bacteria that start to be correlated with the response to some but not to other foods as well. And I will not go further into this for the sake of time. This was also true for the real-time food loggings that we collected, and we performed this study in a real-life situation. And you can see for tens of different foods that the variability in the glycemic response between the individuals is huge, and we can start to see very interesting associations that emerge, including my very personal favorite association on the bottom right. You can see that when we compared the response of people to rice and to ice cream, some people, such as the volunteer on the top, feature, maybe as some of you may be surprised to see, a higher response to rice as compared to ice cream, while the individual on the bottom features the exact opposite response to the same set of foods. And actually, when we looked across our cohort, actually close to 70% of people had a very low response to ice cream, which may be a very good news to many of us. But the most important point, or the most important question that we asked in this study is, can we predict people's individualized glycemic response based on this huge amount of personalized metadata? Now, if we go to the dietitian today, we do get a diet that is based on some predictive power, mainly based on the amount of carbohydrates we have in the diet that we are given. And this has some predictive power. So when we measured it and we calculated it, the predictive power of the gold standard of today is 0.37. And we took it from there, and we took 800 participants with all of this data that we've collected from them, and we developed a machine learning algorithm for each of the individuals that attempts to use all of this data to predict better the response of these individuals to foods that they consumed during the week or did not consume at all. And then we took 100 people that we were completely blinded to, and we took the algorithms that the algorithmic system that we've developed on the 800, we applied it on this new cohort of 100 participants to see whether our system is sane and correct. And these are the results. So the gold standard today, when we go to the dietitian and base our diet on the carbohydrate contents, is 0.37. Now when we start to add personalized features, such as the meal features different people use, we increase the predictability. We add in more of the log features, such as the time of day, sleep, exercise, when we ate yesterday, and you can see that we can dramatically increase the predictability. Now we add in the microbiome features, and we are continuously adding them as we speak to further improve, but you can see that this gives you a whole lot of more predictive power, and we add more personalized features in the form of blood tests and questionnaires, and we get to a much higher predictive power than the gold standard we have today. And what's really cool about this is that we can play around with the different features, and if we only take the microbiome plus the questionnaires, we can basically replace most of the other features and get to almost the exact same high predictability. We then applied the same algorithm that we developed and trained on the 800 people on the new 100-person cohort, and you can see that we could increase it even further from the 0.67 predictive power to 0.70, which is extremely high and actually very close to the limit of prediction, because the intra-individual variability that we do see can bring us to maximum of 0.8, 0.85. So we're not far from the top predictive power that we can actually use. So at this point we were quite pleased with ourselves, but we felt that it is time to put ourselves to the test and to do some prospective evaluation of the new personalized system that we developed. So in this new study, we took a group of 26 individuals, most of whom were pre-diabetic, and we put them through our system. Now, pre-diabetics, as you very well know, are an emerging population. Actually, it is estimated that over 38% of the adult U.S. population are pre-diabetic, and this population would develop, 70% of them will develop full-blown type 2 diabetes within 10 years from their diagnosis. And actually, we have very little to offer them to reverse this tendency. And actually, the dietary recommendations, which me as a physician are trained to give them, when we test them on our cohort, in many of these individuals would drive them faster towards type 2 diabetes. So clearly there is no real solution at the moment for this huge problem. So we took these 26 individuals, we put them through our weekly process, we collected all the data, and we divided them into two groups. Half the people were constructed this personalized algorithm that told them what their individualized bad foods and good foods are, and the other half, we gave all of this data that we collected on these individuals to a group of experts, very experienced dietitians and scientists, who constructed, based on the glycemic responses they saw in the graph, good and bad diets based on what they saw, but not based on the algorithm. And then for 1 week, each of these individuals was given only their personalized good food for an entire week, and then only their personalized bad foods for another entire week, while we extensively profiled them by continuous glucose monitoring, daily microbiota profiling, and all of the rest. Now, I'll give you an example of one of the participants. This lady was given a set of good diets for a week and then a set of bad diets for a week. These diets are only good for her, and you can see an example of one of the good diets and one of the bad diets. And if I would ask you which is the good diet and which is the bad diet, you would probably not be able to guess, because when we did this on our own students that were not involved, we got a 50-50 answer, just like the flip of a coin. In this case, this lady had a good diet that included, for example, ice cream and hummus, and a bad diet for her included sushi and corn. But when we gave her individualized good diet, and by the way, all the good and bad diets in this experiment were isocaloric, so there were no caloric differences between the good diet and the bad diets. When we gave her the bad diet for a week, the bad diet included sushi and corn. You can see that that lady spiked to almost diabetic levels. This is in red. While when we gave her the good diet with the ice cream, you can see in the green graph that she had a flat glycemic response throughout the week, and you can see that the statistical differences between her good and bad week were huge on the bottom. This is another example of another participant, again, very hard to guess. In this case, the good diet included hummus. We think that hummus is one of the healthier diets that we recommend, but also halva and croissant, while the bad diet included fruit. Again, very hard to guess, but you can see that participant's glycemic response throughout the week, and you can see that in the bad week, that person spiked to very abnormal levels, while in the good week, that person was normal, although he started as a pre-diabetic, and again, the statistical differences were huge. And here you can see another participant showing the same responses. Just to show you that I'm not showing you my favorite 3 participants. This is the entire cohort, and you can see that actually in the vast majority of people, we got a very major improvement during the good week as compared to the bad week, although the diets were completely different between different individuals, both the bad and the good diets. And actually, the algorithm-based diets were even better than the gold standard expert diet. And you can see that the prediction in this cohort reached actually 0.8, which is really what we think is the best we can do in this situation. Throughout this intervention study, we sequenced the microbiome every day of those 2 weeks. And what was really interesting is that despite the fact that people started from a completely different microbiome configuration, all of us have a different microbiome composition and function, and despite the fact that each of these individuals was given a personalized good or bad diet, you can see that when most people improve during their good diet, you can identify a cluster of bacteria that dramatically change in these individuals, and another group of bacteria which are correlated to the bad glycemic responses in the bad diet week, despite the fact that these interventions were relatively short-term, only a week at a time. Some of these bacteria that changed, we were pleased to see that were described before us in studies related to obesity and to type 2 diabetes by Jeff Gordon and by others, but many of the combinations and the signatures that we've discovered in this context were not previously demonstrated or discovered, and we are extensively testing them to see whether indeed some of these bacteria that are changing so rapidly in response to diet are indeed drivers of the bad or the good glycemic response. And you can see here some examples of the bacteria which we found, which were described before us in the same exact direction. So what I've tried to convince you in this short presentation is that we have much to learn about this new and exciting axis connecting our diet, our microbiome, and our metabolism, but we are learning more and more, and we can actually now start to utilize the data that we are collecting to use this personalized aspect of our diet in order to perhaps use it as means of diagnosis and therapeutics in metabolic disease. And I will finish by thanking the people who have been doing this really great work in the last 4 years. Many people from my group and from Eran Segal's group are involved. I will also thank the funding agencies which are making this work possible, our clinical collaborators, and I think I'll stop here. Thank you very much.
Thank you, Dr. Elinav. We have time for just 1 or 2 questions. Ivan.
Ivan Rusin, Texas A&M. Thank you so much for a wonderful presentation. I think I reflect the sentiment in the room here by saying that. Quick question. You did mention that you collected or are planning to analyze genetic information on these individuals. You didn't mention anything in the analysis. Is that something that you already looked into or something that you plan to do in the near future?
Yeah, that's a great question, and I should have said it. We've collected from all of the 1,000 individuals genetic information, and we've actually sequenced it using a million SNP analysis. But everything that I've shown you today, all the algorithmic data that I've shown you today, does not include all of this genetic data. What we're doing at the moment is we're taking this as a great opportunity to actually compare head-to-head the genetic predictability power to the microbiome predictability to see whether one of them is better than the other. We have very exciting preliminary results, but these are still preliminary, but it will give us a sense of whether indeed the microbiome, by integrating genetic information into it, is superior in its predictive power, at least in the sense of glycemic response as compared to genetics, and whether genetics could contribute to this prediction.
Thank you.
Eran, this is Marty Blazer. Congratulations on beautiful work. I wanted to find out, since the microbiome was so predictive for you, what were the major predictive characteristics? Were they species or OTUs? Were they networks? Were they genes? What was most informative?
Well, the answer is that there were a combination of them all, and actually there was not one predictor that dominated over the other. I think there were 60-something, if I remember correctly, 60-something parameters in the microbiome which we found to have an impact, a gradual impact on this predictive power. Of course, one needs to mention, and these included both compositional information and metagenome information, mainly in gene and pathway abundance information. And one needs to mention that it really matters what of the data sets you put first into the predictor in terms of the weight of that set of features in making the prediction. But by almost every order that we put, the microbiome was very substantial. And I also need to add that we have collected so much metagenomic data, and there is so much black matter within what we've collected, that we are continuously adding features into this algorithm, and we are increasing the predictability of the microbiome, because we think this would be probably a sensible way in the future of making these predictions without having to put people through this very extensive week of collection. But it is a combination of OTUs and gene, pathway, and even module abundance that give us the prediction.
Wonderful. Thank you very much.
One last question.
Linda Merbaum, Director, NIEHS. Beautiful talk. Question, have you looked for, or in all the information you took about your subjects, did you take any environmental information, such as whether they lived near very crowded roadways, where they were living in relation to kinds of other things, whether they were farming, so you have other information about other kinds of exposures?
That's a great question. Some of this information we did gather, some we did not. Israel is a very small country. Actually, Israel is the size of New Jersey, so we know exactly where people live. We've not gone into this data deeply enough yet, but it's very interesting to find out whether your urban versus village status would affect, or perhaps would explain some of these different features. The other point I'd like to make in that regard is that our population as a whole is very genetically diverse, which makes it a very good hub to study differences and variability, and it contains people from all over the world. So I think there's much more that we can learn from looking better and deeper into the environment in finding new features and new predictions and new insights. We've not done much in that sense yet.
Thank you.
Dr. Elinav, thanks again for a wonderful talk. We really appreciate you joining us today.
It's my pleasure, and good night. Bye.